All right, ladies and gentlemen, we're going to do this video lecture today again. As you can tell, I'm still pretty sick, so here we go. Today we're talking about organic chemistry. So obviously what we talked about last time with the basic chemistry principles is going to kind of lead us into this. This is really where we're going to stay in the biology realm. With the inorganic chemistry, that's really more next year for you. But you had a nice preview, and understanding that stuff is really going to hopefully help you understand the organic part of it. When you're talking about organic chemistry, we're talking about molecules that contain carbon. So that's a really key ingredient there. You've probably heard carbon-based life forms. Now, above that, it says have a backbone made of carbon atoms. So what you're going to notice is, so carbon is really special in the fact that it has 4 valence electrons. That's something you don't really need to know right now. But what that means is that it can form and likes to form 4 bonds. So it can build very long chains in a lot of different directions. And this is really important for biology because biology requires specific molecules, and we can start building these large molecules called macromolecules. You can see up here, macromolecules. It's large molecules. So macro means large. And that's really important for biological systems. And there's 4 major macromolecules that we're going to talk about that are basically essential for all living things. And that's kind of where we're going today. Notice that it says contain carbon atoms for organic compounds. And then you can have all kinds of things bonded, but usually H is in there. It's hydrogen. Oxygen's obviously a big one. And then there's these 3 too. Sometimes we'll have sulfur, nitrogen, and phosphorus. The example I'll give you here is just a basic sugar molecule, C6H12O6. So that's called a monosaccharide. But obviously there's a lot more that we're going to get into here. Inorganic compounds, those are the ones that kind of lack, I don't like the word lack carbon and hydrogen, because there are some inorganic compounds that do have carbon in it. But for the most part, inorganic compounds will lack those 2 things. Example would be something like salt, right? Table salt, NaCl. I don't know, magnesium oxide, MgO2. It's anything that doesn't have that carbon backbone. So this next part here is talking about the difference between molecules, monomers, polymers, and macromolecules. And what's happening here is as you go this way, you're just getting bigger and bigger, right? So molecules here, when we talk about molecules, these are the smallest pieces. I want you to think of these as kind of like Lego blocks. And, we take molecules and we put those together to form monomers. And then we put monomers together to form polymers. And then as we continue to add more and more Legos, we eventually get to the level of macromolecule, which is a functional molecule, okay? So, molecules we talked about last time. We talked about compounds. We didn't really talk about molecules. This is kind of the chemistry nerd coming out of me, but a molecule technically are non-metals bonded together. Okay, so something can be both a compound and can be a molecule. Remember, a compound we talked about has to have 2 different elements, right? So, something like CH4, which is methane, that's a compound, but it's also a molecule because there are all non-metals in there, okay? If we look up here, the example I gave you is sugar, C6H12O6. That's a compound because it's made up of more than 1 element, but it's a molecule because all of those elements are non-metals, okay? This is a compound, NaCl, as it's made up of 2 different elements, but it's not a molecule because sodium is actually a metal. Same thing with the magnesium oxide. So it's not something huge that we're going to test you on. It's going to be bigger next year in honors chemistry. But that's the difference really between molecule and compound. And these molecules, as we start putting them together and start building larger molecules, we start creating what's called a monomer. Now, monomers are more complex than molecules. They're larger than molecules, but they're simpler. So you notice it says smaller, simpler molecules that may bond together to make polymers. So they're kind of more complex than molecules, but they're less complex than the next step, which is a polymer. So as we start putting these monomers together, we build a polymer. And I'll show you all of these things here in a minute. This is just really kind of just the understanding phase. So polymers are larger molecules with repeating parts, those repeating monomers. We've actually already talked about a polymer, and that would be DNA. And no, DNA is actually a macromolecule. If we look at the entire strand of DNA, that's the macromolecule. The monomer of DNA we talked about was a nucleotide. So a nucleotide, remember, is a phosphate bonded to a deoxyribose bonded to a nitrogen base, either adenine, thymine, guanine, or cytosine if we're talking about DNA. So I'll just put nitrogen base here. Okay, that's a monomer. Phosphorus, if we looked at the phosphorus there, that's, sorry, it's not phosphorus, that's phosphate. That is PO3. So there's a molecule, right? Molecule. And when we start putting molecules together, we build a monomer. So the whole thing there is a monomer. So it's obviously more complex than a molecule, but it's less complex than the next step. So if I start putting phosphate, deoxyribose, I'll get more specific now, adenine, and I bond that to another nucleotide on the other side, like so, right? And we even could go one step farther, put another phosphate down here, and I'm starting to add more and more of those monomers together. This is a polymer, because now I'm taking the smaller units and I'm building something bigger. Now, how do we go from polymer to macromolecule? Well, obviously this is not the whole DNA. Like, is this DNA? No, it's just a piece of DNA. If we're looking at the entire strand of DNA, we're talking about, millions and sometimes billions of monomers linked together. That is when we get to the macromolecule stage. Okay, so again, it just keeps getting bigger and bigger and bigger, and we're just building on and building on and building on. All right, so what are the four major macromolecules in biology? Here they are for you. Okay, proteins, carbohydrates, lipids, which are fats, and then nucleic acids. On Tuesday, we are going to be doing lab tests for these macromolecules. We have indicators. These are chemicals that change color or do something significant in the presence of one of these four macromolecules. Okay, so I'm going to give you those lab tests now. For protein, we use something called biuret solution. Now, biuret is blue when it's in neutral, when there's no protein. So I don't know if I should do that here. I'm actually gonna leave that till later, just because there's not a lot of space here, and I don't want this to get really kind of messed up here. So biuret is the lab test. I'll just kind of verbally say it, but when we do the lab on Tuesday, it'll have all of this stuff for you. So biuret is blue when it's no protein. It turns purple, more like a violet, when there is protein present. The monomer of a protein is an amino acid. Okay, and there's 20 different amino acids that we build proteins with. We have an entire section later on in the year where we're gonna talk about proteins and talking about how they're built and so forth and so on. Now the polymer, this is where it gets a little fuzzy. The polymer here is really the polypeptide, right? So if I take an amino acid and I bond it to an amino acid and I bond it to an amino acid, that's obviously, I'm starting to build the polymer. I don't like the word protein here because really when you get to a functional protein, that's really the macromolecule. But again, we're going to spend a lot more time on this stuff later, so that's not really our focus right now. Our focus is just kind of a basic understanding of organization of macromolecules in biology, because it does get pretty complex. We have to start bending these amino acids into a shape for it to turn into a protein. So moving on. The function of proteins, there's a lot of functions of proteins. They're obviously pretty important in biology. They control the rate of some reactions and help regulate some cell processes. These are called enzymes. Okay, so enzymes are proteins. Obviously, they're a building block. They're used to build important things in our body, such as muscles and bones. Those are mostly made of protein. And there are other things. We're going to talk about the cell membrane, and we're going to look at how materials are transported from one side of a membrane to the other. And they're also there to fight disease. Some examples of functional proteins in the human body, we've already covered a couple this unit. Pepsin. So there's a protein that digests protein. Kind of funny. Another one that you've probably heard of before, antibodies, are examples of proteins in the body. And again, we're going to talk about those a lot. Well, pepsin we already kind of covered, but antibodies we'll talk more when we talk about the immune system. All right, next up is carbohydrates. Carbohydrates, we use a solution called Benedict's. Benedict's is blue in color, and when you add it to something that contains carbohydrates, you need an extra step. It doesn't just turn color when you first add it to the solution of carbohydrates. You actually have to heat it, and when you heat it up with Benedict's, it turns a hot color, so red, orange, yellow. Sometimes green. It really depends on the type of carbohydrate that is present. Now, the carbohydrates are actually, so you know what? I made this a little bit too big. Let's move this up a little bit. Maybe make it smaller. Because Benedict's solution is for testing for simple sugars. When I get to the next one, we'll talk about monomers. I'll explain this a little bit more. But this is for simple sugars. So those would be mono and disaccharides. We have another test for starch, and it's Lugol's solution, which is iodine. Oh, I can learn how to spell. All right, we're just going to start over on that word. So Lugol's solution, which is iodine, is used to test for starch, which is a complex sugar. Okay. Lugol's solution turns dark. We actually saw this already when we did the microscope lab. When you add iodine to things that contain starch, you get a really, really, really dark blue. It almost looks black at times, but it is like a really, really, really dark blue. So there's really two tests for carbohydrates, both Benedict's solution for simple sugars and Lugol's solution, iodine, for starch. You might want to just jot that down. This is iodine. It's a solution of iodine. Okay. So let's talk about the monomers. So a monosaccharide is the monomer, and it's just a simple sugar. So we talked about, I already gave you examples, glucose is C6H12O6, right? So that's the monomer and this is true for all carbohydrates. There's actually several different types of simple sugars, monosaccharides. There's glucose, there's fructose, there's galactose. But when I start linking those together, I can start making a polysaccharide. Well, first, if I take two monosaccharides and link them together, C6H12O6, you don't have to write this here because it doesn't really fit. If I put two of them together, that would be a disaccharide, right? There's two. Saccharide in Greek means sugar. So mono would be one sugar, okay? A disaccharide is 2 sugars. Okay, so that would be 2 glucoses together. The chemical formula for a disaccharide, well, let's think about this. 2 times the C's, right? C6 and C6 would be C12. This is where it gets a little weird. When you make this bond, you actually lose a water in that reaction. So we don't go to H24 because water is H2O, right? So if I lose that, it would be H24, but H2O comes out, so then I'm losing 2 H's. So I'm down to C12H22. And since I lost a water, O6 plus O6 would be 12, minus 1, because there's 1 oxygen in water, so O11. So that's our chemical formula for a disaccharide. But I can keep adding on. And every time I add on, I have to remove a water. We'll cover that at a later time. It's called dehydration synthesis. But every time I add another monosaccharide and remove a water, I start to get to a polysaccharide. Okay, and the polysaccharide, again, could be like 3 monosaccharides together. That would be the polymer. So how do I get to the macromolecule? Well, starch is, and cellulose and glycogen, those big carbohydrates, those are thousands and thousands of monosaccharides put together. So again, think back to this. We're just getting from smaller to bigger to bigger to bigger, right? As we go down this, getting smaller to bigger to bigger to bigger, right? The polymer might be 3 monosaccharides together. Technically speaking, the disaccharide is a polymer, right? Because it's more than one monomer. And I'm just going to keep adding on and adding on and adding on, and eventually I get to a big molecule like starch, and that's the polysaccharide. Okay, so the function of carbohydrates is mostly energy. They're a big source of energy in all living things. Okay, there are some structural uses as well. We've already talked about why leafy material is really hard to digest because of that cellulose. Cellulose is a very hard molecule to digest. That's because cellulose builds plant walls. So some plants, all plants actually, because they all have cellulose, but plants, some animals and other organisms also use carbohydrates for those structural purposes. So when we say structural purposes, we're talking about building things, right? So cellulose is used to build cell walls. The examples here were already kind of given, but I guess I'll give like sucrose. Sucrose is table sugar. That's something we haven't talked about yet. That's a disaccharide. So that's made up of 2 monosaccharides. Okay, so its formula would be C12H22O11. Another one would be something like lactose, which I'm sure a lot of you have heard of. That's milk sugar. Oh, struggling again. Milk sugar. So another disaccharide. Okay, so Benedict's solution is kind of going back to here because this is really more pertinent to what we're talking about. Benedict's solution is going to really focus in on testing for the mono and disaccharides. Okay, Lugol's solution is specifically for starch. Right. Okay, moving down. For lipids, these are fats. The lab test we're going to use is something called Sudan 3. So Sudan 3 is soluble in water. It's red in color. And, I think most of us know that lipids don't dissolve in water, right? Think about oil and water, right? Oil's a lipid. They don't dissolve in water. They separate. So what this does, Sudan 3, sometimes it's hard to see within a sample that separation. Sometimes it's difficult to see if there's an actual separation there. So we use Sudan 3 to basically show that separation. You can see it very easily once you add Sudan 3. And you'll get a taste of this again on Tuesday when we do the macromolecules wet lab. The monomer, this is a little bit of a weird one. Okay, it's a glycerol with 3 fatty acids, and it's in the shape of an E. So let me do it this way, actually. I'm going to zoom in here just because it makes it a little easier to write small things. So the glycerol is the backbone of the fat, of the lipid. And then to that, there are 3 fatty acids that are bonded out this way. Excuse me. Oh boy. And there we go. Hopefully now you can see the shape here. It's an E. I'll use the highlighter, right? There's the backbone, okay? And then you have the 3 fatty acids branching out from that. Looks like a letter E. All right, now, that's obviously, again, the simple kind of building block. These can then eventually form bigger lipids. And we'll talk more about lipids another day. So each of these is a monomer, right? Fatty acid, fatty acid, fatty acid, and a glycerol. It's a little bit different than the other ones because there's different fatty acids, there's different glycerols. Not different glycerols, sorry. Glycerol is standard. So this really is the whole, fat. So I really should take this and move that over here because that's the polymer, right? So the glycerol itself is the monomer, fatty acid monomer, fatty acid monomer, fatty acid monomer. When I put them all together, I get a lipid. Now lipids are used for storing energy. So carbohydrates are used for energy. So they're the energy source. But when we, eat too many carbohydrates, we eat too many sugars, or we eat a lot of starch, your body will store that energy in fat. Your body can then turn that fat back into usable energy in the form of glycogen. Remember that glycogen up here? So this is long-term energy storage. Now, there are some lipids that are really important for biological membranes. We talk about the cell membrane later on in the year. We'll talk about something called a phospholipid, which is the building block of a cell membrane. So biological membranes are also really important here for lipids. And for any kind of waterproof covering. So like your earwax is mostly lipid. The reason you have earwax is obviously to trap, here, I'll put it over here, earwax, to trap dirt and other things that get into your ear, but it also is there to make your ears waterproof. Otherwise, they'd be really itchy. Another example of a lipid, a functional lipid, kind of mentioned it. We're going to get into this a lot later on, but a phospholipid is a biological component of cell membranes. All right, for nucleic acids, we don't do a test for nucleic acids. There's no lab test for nucleic acids. We've already covered this because we talked about DNA, but nucleic acids, building blocks, are nucleotides. Remember, that's that phosphate bonded to a sugar and bonded to a base. We can get very specific. With DNA, the sugar would be deoxyribose, the base would be either adenine, thymine, guanine, or cytosine, okay? But that's our building block, and again, I kind of use this as our example above, so I'm not going to get more into it, but the nucleic acid is when I start putting these nucleotides together, forming a small piece would be the polymer. Eventually, I form the full strand of DNA, which would be considered the macromolecule. The job of nucleic acids in living things is to store and transmit genetic or hereditary information. So we've already kind of talked about that as well, right? So you get your DNA from your parents, and it's going to determine traits. So examples here, DNA and RNA. Okay. All right. So in terms of, we're going to get to like building things here in a minute, but reactions are an important part of biology. They're what sustain life. So when we're talking about metabolism, we're talking about chemical reactions. This is not necessarily a biological reaction. This is the combustion of methane. But this is just to understand, what are the reactants and products. So reactants of a reaction are before the arrow, and products are after the arrow. So reactants are what's needed for the reaction to occur, and products are what's made by the reaction. Occasionally you'll see numbers here. These are called coefficients. These basically mean that so there's a 1 here, even though we don't write it, one CH4 molecule reacts with 2 oxygen molecules to make 1 CO2 molecule and 2 water molecules, okay? All right, now in biology, this is just a picture version of that. Actually, that's not that same reaction. That's nitrogen gas, N2, plus hydrogen gas, H2, gives us 2 molecules of NH3. If you want to write that in, you can. Okay, so that's that reaction. There you go. So again, reactants would be these, products would be these, okay? So in biology, we really focus on a special, there's actually a couple processes of building and taking down macromolecules. So we've been talking about and I even started the day off talking about, taking monomers and building polymers. I mentioned down here with the disaccharides that every time I make this bond, right, so turn that into a plus sign. If I'm going to take a monosaccharide and bond it with another monosaccharide to make a disaccharide, what do I get? So that process is called dehydration synthesis. So dehydration, if you think about that, that means losing water. Oh, I guess it's right here. Losing water. So to dehydrate something, we have to take out a water molecule. Every time you make a bond from a monomer to another monomer, we use this process called dehydration synthesis, and we have to remove a water in that process. So monomers are combined or linked to make polymers. And we've already kind of talked about polymers and then eventually the macromolecule. But every time a monomer is added, a water molecule is released in that reaction because there has to be a place for it to bond. That whole process is called dehydration synthesis. Removing water, synthesis means to build. H2O is a product in this reaction. So here's our disaccharide that we just talked about above, okay? This is glucose. You can take my word for it. Every time this is an organic picture. There's actually a carbon there. There's a carbon there. There's a carbon there. There's a carbon there. There's a carbon there. So now if we count the carbons, 1, 2, 3, 4, 5, and 6. So I'm not going to go through all of it, but if I count everything here, this is C6, H12, O6. Okay, that's glucose. And I'm going to add, I'm going to put a little plus sign there. I'm adding a glucose to another glucose, C6, H12, O6. Okay. If you notice right here, this is where the bond is going to form. And notice there's an H there and an OH there. You see that? So in order to make a bond, I have to remove these pieces. I have to remove those pieces. So when I do that, if I take an OH and an H, an OH, I'm just going to just draw this down here for a second. You don't need to. It's a good visual though. If I take the OH out, it's called an OH minus. It's a hydroxide ion. If I take the H out, it's an H plus. And I think most of you know that pluses and minuses like each other a lot. And they come together to form water, right? So that's why it's called dehydration synthesis, because I have to remove the H and the OH there in order to make that bond. If you look on the other side here, now I've got that bond made. Here's that bond right there. Notice the O is left because that's that O right there. See it? So that O is left. And now I've made I've put 2 monosaccharides. This is called a disaccharide. This disaccharide, if I always forget how to spell disaccharide because it's a weird word. If I use glucose plus glucose, I get maltose, okay, as the disaccharide. Now, its chemical formula would be C12, H22, O11. Now, some of you smarty pantses, like I already mentioned above, you're like, wait a minute, Mr. Delbaccio, the law of the conservation of mass says we cannot destroy any kind of matter, right? So we had 6 C's. So we had 12 C's go in, right? We had 24 H's go in. We had 12 6's go in. So where did they go? Well, they're right here, right? So C, we didn't lose any C's in that process. So C12, we lost 2 H's in the water right here. So that's H22. We lost 1 O, O11. So this is the disaccharide, plus we get 1 molecule of water. For every bond you make, you're going to lose a water. So if I wanted to add another glucose on here, I would get another water molecule. Okay. We talked about sucrose being a disaccharide. A sucrose molecule is actually a glucose plus a fructose. So we can add that one here, glucose plus fructose. So these each would be C6H12O6. Those are both monosaccharides. Gives us sucrose, that's table sugar, plus one water molecule. Okay. Milk sugar is actually glucose plus galactose, gives us lactose plus water. Now, your body is able to go the other direction too. Obviously, when you eat carbohydrates, so you eat something like maltose, it wants to be like, hey, wait a second, I don't want these in twos because that's harder to break down and use for energy. So it's going to want to break that bond. Hydrolysis is the reverse process. So hydro means water. Lysis means to break. So there's a couple ways to look at this. I like to think of it as using water to break bonds. Right? So in order to digest macromolecules, we have to be able to break those bonds. But we can't just break them. I can't just be like, I'm going to rip you apart and take you apart. I have to put back what was taken out to make that bond. Remember, water was used to make that bond. So I have to put that back in. I have to take the water, I have to change it back into H plus and OH minus, and I got to reinsert those back into these molecules. Otherwise, they wouldn't be glucose again. So that is called hydrolysis. It's the use of water to break a bond. If you take a look at this whole equation now, get rid of all that. You've got the disaccharide, C12, H22, O11, and we add that with water because I have to take the water and put that back in. Water's H2O, right? And then I get two glucoses back out, C6, H12O6, C6, H12O6. Okay, so this is the reaction in formula form. Okay, so hopefully that's a kind of a good intro here. We're going to spend a lot of time on this stuff. We're going to talk about dehydration synthesis a lot. We're going to talk about hydrolysis a lot. So the nice thing is that these two processes don't change. It doesn't matter what macromolecule we're building or taking apart. If I'm building it, I'm using dehydration synthesis. If I'm taking it apart, I'm using hydrolysis. Like if I want to build a lipid, just one more example. If I want to build a lipid, I've got glycerol. And I've got 3 fatty acids, right? If I want to build this lipid, take that fatty acid, and I got to make a bond right here. Got to make a bond right there. I'm not at a lipid yet, but if to make that bond, what do I have to take out? Water. That's dehydration synthesis. If I want to take this fatty acid now, make a bond right here. Again, I'm just building a lipid here. I need to take out a water. And if I want to take this fatty acid and I want to make a bond right here, I need to take out a water. So I'm going to give you a second. I want you to actually write out the actual reaction to make this glycerol. So what would I need as a reactant? And then what would I make in the end? I'm gonna give you about like 15 seconds. Let's see if you can get it before I write it down. I'm not gonna do the second part of the song. Here we go. So what do I need? In order to build this lipid, I needed a glycerol plus 3 fatty acids. That's my reactants. My product would be a lipid, but not a lipid by itself, plus 3 waters. Dehydration synthesis. What would hydrolysis look like? The exact opposite. So in order to break those bonds, all I have to do is switch my arrows around. So in order to go through hydrolysis, we can label this dehydration synthesis. In order to break those bonds, I have to put the water back in. And then I can get the 3 pieces back out, right? I can get the glycerol and 3 fatty acids back out. So again, it's just flipping this equation around. If I want to break that lipid, I have to add 3 waters to it. And then I'll get the individual components back out. Just abbreviating. This is hydrolysis. All right, so the good news is, since it is a recording, you can watch this over and over and over again. You can ask questions. This is tough stuff, I get it. It's the first time you've been probably exposed to organic chemistry. So what we're gonna do now is we're gonna work on and finish up the rest of that macromolecules activity. You're gonna use this kind of stuff in that activity, and I know you did some reading on this stuff for homework. So hopefully with all of what we just talked about, you have a pretty good handle on it. On Tuesday, we're going to be doing the macromolecules wet lab. So we're actually going to be doing these tests in class so that we can start to see how to identify proteins, carbohydrates, and fats. Remember, there's no test for nucleic acid. All right, that's all I've got for you. So we're going to go ahead and move on to the macromolecules activity.